Hello students, welcome to third lecture of chemistry in everyday life. In previous lecture we discussed about neurologically active drugs and now let's talk about the second type. So today's topic antimicrobials. So it will act against the microorganisms. Diseases in human beings and animals may be caused by variety of microorganisms such as bacteria, virus, fungus and other pathogens. So all of these we know that are some or less can cause illness or different type of diseases. Certain type of microbials are also useful for us, that means they will help us in certain type of reactions and they can even use for medicine production, but majority of them can cause diseases. An antimicrobial tends to destroy or prevent development or inhibit the pathogenic action of the microbials such as bacteria, which is known as antibacterial drugs, fungus, antifungal agents, virus, antiviral agents, or other parasites, anti-parasitic drugs selectively. Antibodies, antiseptics, and disinfectants are antimicrobial drugs. So all of them acts differently and their uses are also different. So let's talk about the antibiotics first. So antibiotics are used as a drug to treat infection because of their low toxicity for the humans and animals. So that means they are toxic for bacteria or microorganisms, but they are less toxic for us. But some or less they are toxic for us. So we should use it in proper manner. Initially, antibiotics were classified as chemical substances produced by microorganisms. That means we are using microorganisms to stop microorganisms. Bacteria, fungus, and molds, they are helpful in the production of earlier antimicrobial drugs, and that inhibits the growth or even destroy the microorganism. The development of synthetic methods has helped in synthesis of some of the compounds that were originally discovered as product of microorganism. That means initially we were obtaining them from the microorganism, but later because of developed science we are able to produce these type of chemicals in laboratories. Also, some purely synthetic compounds have antibacterial activity and therefore definition of antibiotics have been modified. An antibiotic now referred to a substance produced wholly or partially by the chemical synthesis which in low concentration inhibits the growth or destroys microorganisms by intervening in their metabolic processes. The search for the chemicals that would adversely affect the invading bacteria but not the host began in 19th century. That means before that initial antimicrobials were also attacking host. That means they are dangerous for us as well. But overall effect of these type of antimicrobials on the body were comparatively less than bacteria or microorganisms. That's why we were using it. Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated the arsenic based structures in order to produce less toxic substances for the treatment of syphilis. He developed the medicine arsenophenamine known as salvarsan. Paul Ehrlich got Nobel Prize for the medicine in 1908 and it was the first effective treatment discovered for syphilis. Although salvarsan is toxic to human being, its effect on the bacteria spirochete which causes syphilis is much greater than on human beings. That means it was comparatively less harmful than the bacteria which causes this type of problem in the body. That's why although medicine is also damaging us, we were using it in earlier times. At the same time, Ehrlich was working on azodize also. He noted that there is a similarity in the structure of salvarsan and azodize as AS double bond AS linkage present in the arsenophenamine resembles the N double bond N linkage present in azodize in the sense that arsenic atom is present into place of nitrogen. That means in arsenic compounds we have double bonded arsenic atoms just like in nitrogen azo compound we have double bonded nitrogen elements with each other. So we got an idea to replace arsenic with nitrogen, which is less toxic or non-toxic for us. He also noted tissues getting colored by the dye selectively. Therefore, Ehrlich began to search for the compounds which resembles in the structure to azo-dyes and selectively bind to bacteria. So, as mentioned here, certain type of dyes are used to color bacteria. So, let's say you want to observe bacteria under the microscope, but their structures are not clear because their inner parts are not easily visible in under the microscope. So that's why we use specific type of inks or dyes which can color the bacteria. So we will add some drops in the culture or our sample which can colorize the inner structure of bacteria. That means these type of colored compounds will bind with the bacteria, but these type of compounds are not hazardous for them. But on the basis of this point, that colored compounds can combine with bacteria if we can add certain type of drugs, if we can attach certain type of drugs with this type of dyes, then those drugs will attach with bacteria as well along with dye, and they can kill or prevent those bacteria. And all of this structure which we are discussing are mentioned here so that you can compare and you can see yourself. So as we can see, salvarsan and azodie mentioned last, they share similar type of structure and similar type of bondings. Arsenic double bond arsenic. Similarly, in azodie we have nitrogen double bonded with nitrogen, and then both of the nitrogens are attached with aromatic rings, just like in salvarsan arsenic is attached with rings. So in 1932 he succeeded in the preparing the first effective antibacterial agent, Prontosil, which resembles in the structure to the compound salvarsan. Soon it was discovered that in the body Prontosil is converted into the compound called sulfanilamide, which is the real active compound. So that means Prontosil was not actual drug, but it will convert into sulfanilamide and then it will act against the microorganism or diseases. So that's why we finally found sulfa drugs. So sulfa drugs were discovered and large range of sulfonamide analogues were synthesized later. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the bacterial properties of Penicillium fungus. Isolation and purification of active compound to accumulate sufficient material for clinical trial took 13 years. That means after the discovery of Penicillium fungus against the antibacterial bacteria, that means antibacterial property, we actually got that compound for practical use 13 years later. Antibiotics have either sedal or killing effect or static or inhibitory effect on the microorganism. That means either these type of drugs will kill the bacteria or they will make them unconscious, that means inactive, so that they cannot cause harmful effect inside the body. So we have mainly two types of antibacterial or antibiotic drugs. So that means suppose we are using static type of antibiotics, so you have to complete the proper doses of the antibiotics drug. Otherwise, bacteria are not completely killed inside the body. They are present but they are unconscious. Suppose that bacteria can live up to, let's say, 7 days in unconscious conditions. So doctor normally gives 8 or 10 days of medicine so that those bacteria can be killed in their unconscious form. But if you take medicines for few days and bacteria are unconscious, that's why you do not feel any harmful effect of those bacteria inside the body, and we stop taking the pills or medicines. Let's say after 4 days you stop taking the medicine, but bacteria are unconscious, they are not killed or removed from the body. Then they will become conscious and then they will start their harmful effect again. That's why doctors specifically inform us to complete the course of the medicine. And this is the reason, if you do not complete the course, then bacteria will revive again, and then we need even stronger medicine to fight against them. And second type of drug, as mentioned here, killing effect. So those type of drugs are also harmful for us. So they are not advisable if there is not extreme condition. That means we should take static type of antibiotics, because sedal type of antibiotics are hazardous for us, or they are hazardous for some useful or friendly bacteria which are present inside the body. Certain bacteria are present inside the body which helps in the digestion of food or absorbing the uh, nutrition portions of the food. So few example of two types of bacteria or antibiotic are mentioned here. So bactericidal, that means which can kill the bacteria, penicillium, aminoglycosides and afloxacin. Then bacteriostatic, that means they can inhibit the growth or make them unconscious, erythromycin, tetracycline and chloramphenicol. The range of bacteria or other microorganisms that are affected by a certain antibiotic is expressed as its spectrum of action. Antibiotics which kill or inhibit the wide range of gram positive and gram negative bacteria are said to be broad spectrum antibiotics. And those effective mainly against either gram positive or gram negative bacteria are called narrow spectrum antibiotics. If effective against the single organism or diseases, they are referred as limited spectrum antibiotics. Penicillium G has narrow spectrum. Ampicillin and amoxicillin are synthetic modifications of penicillins. These have broad spectrum. It is absolutely essential to test the patient for sensitivity or allergy to penicillium because before it is administered. In India, penicillium is manufactured at the Hindustan Antibiotics in Pimpri and in private sector industries. Chloramphenicol, isolated in 1947, is a broad spectrum antibiotic. It is rapidly absorbed from the gastrointestinal tract and hence can be given orally in case of typhoid, dysentery, acute fever, certain form of urinary infections, meningitis and pneumonia. Vancomycin and ofloxacin are the other important broad spectrum antibiotics, and antibiotic dicydazarine is supposed to be toxic towards the certain strains of cancer cells. So general structure of penicillin and chloramphenicol are mentioned here, and as we can see they are very complex structures. Then we have second type, antiseptics and disinfectants. Antiseptics and disinfectants are also chemicals which either kill or prevent the growth of microorganism. So these chemicals are for external use only. Initially, first category was a drug which should be injected or we should swallow the pills, and then they will act against the microorganism inside the body. And these are for surfaces, that means either for wounds or to clean the surface or skin, or for non-living substances like floors, tables, etc. So antiseptics are applied to the living tissues such as wounds, cuts, ulcers and diseased skin surfaces. That means for external use for living organism. Example are furacin, soframycin, etc. These are not ingested like antibiotics. Okay, so important point, these are not ingested like antibiotics. Commonly used antiseptic, Dettol is a mixture of chloroxylenol and terpineol. Bithinol, the compound is called also called bithionol, is added to soap to impart antiseptic properties. Iodine is a powerful antiseptic. We still use iodine as antiseptic to clean wound from microorganism. So its 2 to 3% solution in alcohol water mixture is known as tincture of iodine. It is applied on wounds. Iodoform is also used as antiseptic for wounds. Boric acid in dilute aqueous solution is weak antiseptic for eyes. So structure of chloroxylenol and terpineol and bithionol are mentioned here. Disinfectants are applied to inanimate objects such as floors, drainage systems, instruments, etc. Some substances can act as antiseptic as well as disinfectant by varying the concentration. So generally antiseptics are mild and disinfectants are strong compound. For example, 0.2% solution of phenol is an antiseptic while its 1% solution is disinfectant. Now you may ask why we do not use higher concentration for cleaning these wounds. That means why shouldn't we use 1% solution of phenol for antiseptic purpose? Because higher concentration can damage the tissues. That's why we should use lower concentration of the chemicals as antiseptics and higher concentration as disinfectants. Chlorine in the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide in very low concentrations are disinfectants. That means all of these chemicals mentioned can be used for cleaning surfaces or floors and make them free from bacteria or microorganisms.